So I'll be talking about tools for vaccine development, and I'll be focusing primarily on my tools because that's what I know. And I know that there are many tools being developed also in Korea, so I look forward to discussing with you about those tools. I want to thank the organizers for inviting me to Korea. It's a pleasure to be here. So what I'm going to be talking about is really rapidly over the course of the next 25 minutes is how we identify T-cell epitopes for vaccine design, we stack the deck for immune response, we can actually design vaccines based on that T-cell epitope content. I'll be talking about a few tools there and how we're focusing on developing broadly covering vaccines for influenza and COVID and pan-coronavirus. We also are very concerned about antigen design because we have discovered that pathogens can camouflage their antigens from immune response. And I'll talk about how we can engineer those antigens to improve the immunogenicity by removing tolerogenic epitopes. So the tools we've developed are listed here, and I'm not going to talk about several of them. So I'm just going to really focus on the two at the top, IVAX and EPIC. IVAX is used to design vaccines, and EPIC is actually a more recent tool that's now being used commercially that can predict the efficacy of existing vaccines against new circulating strains. So our work has been funded by the NIH and Department of Defense, and the reason I'm putting this slide up here is just to show you the broad applications. Not only do we work in the virus space, but we also work on bacteria. We have been focused also on parasites as well. In the past, we've worked on malaria, so it's nice to hear a malaria talk today. The vaccines that are currently being developed using our technology include a malaria vaccine that was funded by USAID, a vaccine for pan-coronaviruses, which is using OMV particles for delivery, being developed by Intravac, and that's funded by CEPI. We also have a flu A vaccine, which is a universal vaccine that is being developed at St. Louis University by Dan Hoft. Zoetis is actually using the EPIC tool in a commercial program for identifying the best PCV2 vaccines for swine. And we have been working on a COVID vaccine, which was actually an epitope-based vaccine, and it was under consideration for further development by NIH until the current president took over. So now it's no longer in consideration for development at the present. So IVAX is a tool that I wanted to talk about today, which is a platform for designing vaccines. It is web-based, and it can be used, it's based at our location, but it can be used internationally and has been used internationally by scientists who are developing vaccines. We also develop vaccines in collaboration. The concept is to make a seamless vaccine design program, and to my knowledge, this is the only one that actually exists currently. So the concept that we've been working on for 25 years plus is that we can go directly from pathogen sequences to vaccines. So whatever the pathogen is, including cancers, we can decipher the key element, and to me, the key element actually is the T-cell epitope, and that's what's illustrated here. You can see that those pathogens or cancers can get broken down into peptides that actually then get processed and presented at the surface of the antigen-presenting cell. If you can sequence the pathogen, you can identify the T-cell epitope that may be contributing to protection, and that will actually be something that you can put in a vaccine and use any platform whatsoever. So we are platform agnostic. Now, we heard a lot about antibodies today in the influenza talk, and I want to remind everyone that T-cell epitopes are contributors to protective immunity from vaccines, and a great example of that is actually the 2009 swine influenza, the swine origin influenza so-called pandemic, where we saw that if you had pre-existing immunity to this previously circulating influenza A strain, you were protected against the strain of the new strain, pandemic H1N1. And it was through T-cell epitope conservation that protection was provided, since the antibody targets were not conserved between H1N1 and the previous strain. So we now know that T cells are significant contributors to protection. So that's why we've been focusing on them. And I want to mention that we don't just make T cell epitope-based vaccines, but we also make whole antigen vaccines where we engineer in better epitopes. Why is it important to include T cell epitopes? It's because they actually control antibody development. Why do you have high titer antibodies to influenza? Is because there are T cell epitopes that are driving that B cell response and generating higher titer and higher affinity antibodies. There are also Tregs that are present in the lymphoid follicle that can restrain the development of those antibodies, and those Treg epitopes are important to discover and remove from vaccine antigens to make better vaccines. So first I'm going to mention how we identify T cell epitopes. Basically, it's fortunate for us the T cell epitopes are linear, so we can simply look at the sequence and identify class 1 and class 2 epitopes. This has now become pretty common. Lots of different tools exist. Our tool is called Epimatrix. We have also pioneered the concept that the more T cell epitopes you have in an antigen, the more likely you are to drive an effective immune response. So we count up the epitopes and then determine whether the antigen has potential as a vaccine. And here on this slide, you can see that there are two antigens. One has many epitopes, one has fewer epitopes. And then if you basically line them up on a scale, you can identify the one that's going to be the most immunogenic. Why is this relevant? Well, it turns out good vaccines have high T cell epitope content in the red zone. You can see hepatitis B surface antigen, a very effective vaccine driving high antibody titers. Tetanus toxin, a little bit lower, also a good vaccine. But you see two candidate antigens that have failed in the clinic. One is a MTB antigen that failed in the clinic, and the second one is the CSP antigen. And so effective vaccines on our scale, or effective antigens, would be high on the scale, and poorly effective antigens would be low on this scale. The two examples that I'm just pointing out here are the RTSS CSP vaccine, which actually has had pretty poor efficacy, only 4% over time, and then the MVA-85A, which failed in the clinic and potentially due to low immunogenicity. So humans are not mice, and we're looking at human immunogenicity in that particular example. So we can also identify regions of immunogenicity. So we use a tool called Cluster to stack the deck for immune response. What I mean by that is we can look through a protein sequence and identify regions that contain T cell epitopes that will bind promiscuously to different HLA. And this is a technique that we use to identify epitopes that can drive an effective immune response and to potentially remove those regions of the protein that don't actually contribute. So whether you make an epitope-based vaccine or just simply remove long sections of the antigen that are not effectively driving an immune response, that's one way of doing that. So Cluster can identify these clusters, and then we can assemble them into what we call an immunogenic consensus sequence by identifying regions. I don't know if this is actually working. Nope. By identifying regions shown here in red that are highly conserved across viral strains, and then adjacent regions that may be conserved in some of the strains but not all of the strains, and we sew those together into one consensus sequence that can actually be used to drive effective immune response. The example is with Zika. We worked with Dan Hoft and Chris Eickhoff at St. Louis University, and you can see here on this slide that we use the immunogenic consensus sequence approach, and the immune responses highlighted in 4 donors by the blue arrows actually show our epitopes and our immunogenic consensus sequence epitopes versus the other epitopes which were identified using the overlapping peptide approach. So obviously much better immunogens in vitro in patients exposed to Zika, as shown here. We're actually working on 2 vaccines with Dan Hoft. One is the influenza vaccine. That's a universal influenza vaccine for influenza A. The other one is the Zika vaccine, which apparently we have Zika in the United States now, and it's a problem. Correlation studies, I'm not going to go into the detail, but we have quite a lot of validation showing both T-cell epitope response and HLA binding responses for epimatrix. We use binding assays to confirm the predictions, and here you can see we get 90% correlation with the predicted epitopes and in a wide range of HLA-DR alleles. So other tools that we use for designing vaccines that may be of interest to this audience include Conservatrix, which we first developed for HIV, where we can look at different sequences from different clades, for example, and different quasi-species and identify highly conserved T-cell epitopes simply using the linear sequence of the protein. So that's a great tool for variable pathogens such as hepatitis C, Zika, COVID, et cetera. So we use Conservatrix quite a bit. EpiAssembler, I already mentioned, produces those immunogenic consensus sequences, and then VaccineCAD puts the epitopes together in a sequence without having pseudo-epitopes at the junctions. And here I've used words to illustrate how if you put 3 epitopes together, you can create new epitopes at the junctions, so you actually have to remove those by reordering the epitopes. And we have a tool that does this iteratively in silico. So lastly, I wanted to, well, not lastly, but one of the other features that I wanted to talk about is the idea of determining whether you have a vaccine that may already work against circulating strains. And this is a problem with, especially with COVID, as I'll show you. So we recently developed this tool, which is called EPIC. It compares T-cell epitopes across strains. And in this radar plot, the green line is a vaccine that has broad coverage as you go around the radar plot against all the different strains that are highlighted in this plot, except for the ones at the very bottom right. The blue line illustrates a vaccine that does not have good coverage. And so obviously, if you were going to select a vaccine to try to get the broadest coverage, you would select the one with in the green line that has the broadest area under the curve, so to speak. This is actually something that has been used to compare the different viruses, and the example here is the H1N1 pandemic. We found when early on in the 2009 pandemic that there was high conservation in the T-cell epitopes between the Brisbane circulating strain the year prior that was also in the vaccine the year prior, and H1N1. And we predicted actually that there would be good protection against H1N1 pandemic influenza from exposure or vaccination. And that was subsequently shown to be true in studies carried out by Peter Doherty's group and Richard Webby, and they showed actually that the exposure to Brisbane was protective in ferrets. Now, the applications for EPIC are important because we can actually first identify a vaccine that confers the broadest cross-reactive cell-mediated immunity in an outbreak, especially when the virus is under high immune pressure and is evolving rapidly like COVID. We can also understand how viruses evolve in response to immune pressure. We're looking at that with pig influenza and showing that actually the immune response that is generated by vaccines causes viral evolution. We also have developed websites so that companies can select the best vaccine for circulating strains in swine herds. That's the CircoMatch website. And we can also define when vaccines may start to fail against new strains of viral pathogens. And so here's an example where we actually measured the predicted coverage of 2 different vaccines shown in blue and red had the higher EPIC score, and in this particular example, it was a combination of 2 different strains. As Ian Barr was talking about, maybe 2 different strains of the same pathogen may give you broader, in this case, T-cell epitope coverage. That's what we're observing here. And we did actually see in the field observation, that the predicted protective coverage of the orange vaccine was actually confirmed. And that's why Zoetis now uses this EPIC tool in the form of CircoMatch as a website for their veterinarians. In the case of COVID, it's really interesting. If you look at green, the green line, that's actually the Wuhan original vaccine in COVID. And we got pretty good protection by either exposure or vaccination with the Wuhan strain up until the emergence of the Omicron virus, at which point you see that protection from Wuhan dropped off and a new strain of virus emerged, which was actually Omicron. It took us until much later, probably I think it was almost a year later, to actually develop the vaccine, at which time we started putting pressure on Omicron, not just in the form of immune responses to Omicron, but also vaccine responses to Omicron. And what we saw after that was that once we developed the vaccine, which is the orange line there, that the T-cell epitope coverage dropped off rapidly and new strains emerged. So we then developed an XBB vaccine, and then immediately the pressure on the virus caused the emergence of yet another strain. We have developed a T-cell epitope-based vaccine that is shown here in the dotted black line, which actually would have been a pretty good vaccine all the way up until XBB. And then if we added one epitope to the vaccine, we would have covered had better coverage, getting us above that 85% coverage line, which we think is what is required for protective efficacy against variant strains. So unfortunately, that vaccine is not under development due to the current scientific climate in the United States. Just briefly about personalized vaccines and individual immune responses, I found this paper by Greg Poland so interesting because he illustrated in a genome-wide survey of genes which genes were providing a protective immune response, and it turned out that it was HLA-DR. And so having a strong immune response, you had the right DR alleles to present the measles vaccine, and a weak immune response was related to low immune response with low T-cell epitopes presentation by the HLA of the individual. So this is a genome-wide survey that actually demonstrated that T-cell epitope presentation was critically important for the protective effect of measles vaccine. So we've actually looked at this, and we've developed a tool called ITEM, which I won't have time to present today, which allows you to weigh how many T-cell epitopes are actually presented by each individual in a population, and to predict the efficacy of a vaccine for an individual person. Now I'm going to talk about correcting antigen design for immune camouflage. The concept here is that vaccines or pathogens have actually developed means of escaping immune response. So when we're looking for T-cell epitopes, we're obviously prioritizing epitopes that are cross-conserved across strains because that may provide protection against the next infection. We're looking for epitopes that are not conserved with the human proteome because we think that those are going to be the most effective T-cell epitopes, and we're trying to avoid epitopes that are highly cross-conserved with the self-proteome, not only because of potential adverse responses, but also because we want to avoid the induction of tolerance. And here what I'm talking about is that the epitopes that are predicted by computer tools are generally binding the same to HLA, but what's different about those that turn on a T effector response versus a regulatory response is the T-cell receptor face. And so here we've started to look at this and understand that if there are self-like epitopes present in pathogen sequences, that will reduce the immunogenicity of that vaccine. So we've developed a second tool which allows us to look at the T-cell receptor face. So Epimatrix looks at the HLA binding, and what we're asking is, what does the T-cell receptor see? We isolate those amino acids, and then we start looking for conservation in the human proteome. And that's illustrated here. The green square is a pathogen protein. The blue squares are different epitopes in the pathogen protein. You can see the one at the top right has an extensive cross-conservation with the human proteome, whereas this one at the bottom left has very limited cross-conservation with the human proteome. And we've shown in numerous studies that there's a significant difference in the immunogenicity of these two types of epitopes, and that the one at the top can actually be tolerogenic. So that was published in 2013, that observation. Here are 2 example epitopes. One's an envelope, highly cross-conserved across all HIV clades. This epitope in envelope is tolerogenic in vitro. Same thing with hepatitis C. This is an NSP7 protein epitope that's highly conserved across the subtypes of hep C, also tolerogenic. We've actually looked at this in malaria vaccines. I'm going to skip this. So, sorry, just to mention that we've improved the algorithm, the Janus matrix algorithm, by introducing machine learning to identify epitopes that are conserved in different subgroups of proteins in the human body, including looking at thymic expression, circulating proteins in plasma, as well as proteins that are highly conserved in different compartments. And so that actually has led us to be able to more accurately identify tolerogenic epitopes and discriminate those from T effector or immunogenic epitopes. The examples provided here are the epimatrix alone, epimatrix with Janus matrix 1.0, and epimatrix with Janus matrix 2.0, where we get a much higher correlation between the T cell epitope, effector and regulatory content, and immunogenicity. So again, can we actually predict which patients will respond to a malaria antigen? And here what we've done is we looked at the potential for binding, shown in blue, and you can see clearly, well, maybe not so clearly, but across the top are the HLA-DR alleles. Some people will present many of the epitopes from this particular protein, and some people will present few. That's represented in these bars. The people who present more epitopes have a higher immune response in vitro than the ones that don't present the epitopes. If you add the Janus matrix analysis, you get a much higher correlation between immune response to those epitopes and the outcome in the T cell assay. So we've used this with the CDC to discriminate potential Bordetella pertussis antigens. We've eliminated antigens that were potentially tolerogenic and selected with them 64 antigens that they're developing further for pertussis vaccine development. Lastly, I just want to mention that we can engineer antigens. H7N9 was interesting to us because we predicted that it had a low T cell epitope content and would not be very immunogenic. And it also contained a regulatory T cell epitope that we engineered to introduce a more immunogenic epitope and remove the tolerogenic content. What we actually demonstrated is by simply changing 3 amino acids in the HA, we were able to improve the immune response by 5-fold in terms of the antibody titer and by 20-fold in terms of the B cell response. So in summary, hopefully I've illustrated for you that in silico tools can be employed to predict both immunogenicity and tolerance, that self-proteins can drive both effector and Treg responses. That's important for autoimmunity. But pathogens have learned to use those self-like epitopes to avoid immune response. And so we can actually remove those self-like epitopes to improve vaccine antigens. We also work on biologics, so the immunogenicity risks for biologics and also for vaccine design can be personalized. So that's something that I didn't talk about, but we're working on personalized vaccines for cancer. And in silico tools can identify and fix poorly immunogenic vaccines. So these are the papers about the tools that I've described. Most on the right is the IVAX platform. On the left is the ISPRI platform, so those are available to you. Here are the people that make it happen. And I won't have the time to introduce each one of them, but they all have contributed to the success of this platform. And thank you very much for your attention.
Thank you very much for your great talk. I think we can take maybe a couple of questions. Yeah, Professor Lee.
Congratulations, Annie.
Thank you,
for
Jim.
your, non-stopping expansion of new pipelines at Epivax. I have very small, specific question about, T cell epitope, cluster prediction. The fate of T cell epitopes is, the processing by proteasome and presented in the context, presentation in the context of image class 1 molecule. Why should they, cluster to have, better responses?
Why do
as
they
you
cluster?
predicted? They should be, cut.
Well, when you talk about HLA class 1, actually, HLA class 1 epitopes are scattered. What's interesting about HLA class 2 is that they have similar amino acid side chains for binding into the HLA binding pocket. So generally, they can cluster because the side chains that are used for each of the different HLA-DR alleles are somewhat similar. And that's why you see those regions, which are very useful for making vaccines and also for modifying vaccines. You can make a single amino acid change in the cluster, and it will affect almost all of the HLA-DR alleles that present that epitope.
Then, you could, engineer the antigen. If you identify the, T-cell epitopes, then, you could, cluster them into a certain place. Then you will have better responses.
Yes.
Am I right?
Yes. And you can introduce T-cell epitopes, modify T-cell epitopes by increasing their potential for binding or by reducing the TCR-facing residues that contribute to tolerance. So that's an engineering aspect of our work, and we are actively doing that.
Thank you.
Okay, Professor Barr.
You've gone to a lot of trouble to remove the epitopes to avoid Tregs. Have you looked at doing the opposite and using Treg epitopes for autoimmune therapies?
Well, I just came from a conference in Japan where we talked only about Treg with Professor Sakaguchi, who really discovered Tregs. And actually, we are working very hard on introducing regulatory T-cell epitopes into whatever, you name it, drugs that are being used for therapeutics, but also to promote the use of Tregs as a means of treating autoimmune disease. Thank you. That's a good observation.